Good morning. I'm Dr.S.Selvakumar, Associate Professor at Department of Agricultural Biotechnology, Bharat Institute of Higher Education and Research. Today, we are going to discuss about U18PCAC401, plant molecular biology. First, let us discuss about introduction to the basic molecular biology. What is molecular biology? Molecular biology is the branch of biology and that studies the composition, structure, and interactions of the cellular molecules such as nucleic acids and proteins. So that carry out the biological process essential for the cell's function and the cell's maintenance. So basically, molecular biology plays the important role in understanding the formations, actions, and regulations of various parts of cells, which can be used to efficiently target the new threats, diagnosis, diseases, and understand the physiology of the cells. It's called molecular biology. Next, I'm coming to what is the importance of the field of molecular biology. So, molecular biology plays the important role in understanding the formations, actions, regulations of various parts of the cells, which can be used to efficiently target the new drugs, etc. And it has supported and extended our understanding of evolutionary relationships based on the traditional anatomy and the heritability and variation in the traits are essential parts of the Darwin's theory of evolution by the natural selection. So molecular biology has clarified the nature of genes and the sources of the variations. Next, we'll come into the next importance of the molecular biology. So, the molecular genetics utilizes the laboratory tools of molecular biology to relate the changes in the structure and the sequence of human genes to the functional changes in proteins or protein functions, and ultimately to health and disease. So molecular medicine is a wide field. It's a different field. Wide field helps to understand the normal body function and the disease pathogenesis at the molecular level by using physical, chemical, biological, as well as medical techniques with the goal to develop the molecular medications to correct the dysfunctioned genes. Next, I'm coming to with the development of molecular biology, some DNA-based technologies have shown the great potentiality in promoting and the efficiency of crop breeding programs and protecting the germplasm resources and improving the quality and outputs of the various agricultural products and protecting the eco-friendly environment, etc. Next, coming to, so what is the basic differences between molecular biology and cell biology? So molecular biology is the branch of science that considers the nature and science of living organisms at the molecular. Whereas the cell biology typically more focused on the structure and functions at the cellular level and not necessarily the genetic impact on the larger organisms. Next, I'm coming to the subdisciplines of molecular biology field. What are the subdisciplines? So, comparative genomics. This is the study of human genetics by comparisons with the model organisms such as rats, mice, fruit fly, bacteria, fungi, etc. Next, we're coming to DNA forensics or forensic medicine, functional genomics, gene therapy, genomics, and pharmacogenomics, proteomics, and these are the sub-disciplines of molecular biology. Next, I'm coming to prokaryotes. What is prokaryote? Prokaryotes, that is, any organism that lacks a distinct nucleus and other organelles due to the absence of the internal membrane. Bacteria are among the best known prokaryotic organisms. So the lack of internal membranes in the prokaryotes distinguishes them from the eukaryotic organisms. So what are the examples of the prokaryotes? So there are two different types of prokaryotic domains. One is called bacteria, second one is called archaea. So what is bacteria? What is archaea? So the bacteria and archaea split from each other early in the evolution of the life. So bacteria are very diverse. It's ranging from disease-causing pathogens to beneficial photosynthesis and symbiotic. Whereas the archaea are also diverse, but none are pathogenic and many live in the extreme environments. Next, I'm coming to the eukaryotes. So eukaryote, any cell or organism that possesses a clearly defined nucleus. Underline clearly defined nucleus. So the eukaryotic cell has nuclear membrane that surrounds the nucleus, in which the well-defined chromosomes. What is chromosome? The bodies containing the hereditary materials are located. Next, I'm coming to the eukaryotic cells also contains organelles including mitochondria, Golgi bodies, endoplasmic reticulum, and membranes, lysosomes, etc. Next, I'm coming to the examples of eukaryotic organisms. So, single cell organisms like yeast, paramecia, amoeba are all the eukaryotes. In case of plants, grasses, potatoes, and pine trees are all eukaryotes. As are algae, mushrooms, and tapeworms, and of course, fruit flies, and also the examples of the eukaryotes. So, eukaryotic cells may be classified two different groups. What are the two different groups? And based on the number of cells that make an individual organism. Number one, the unicellular eukaryotics and multicellular eukaryotic cells. So unicellular eukaryotic cells, the first category. The second category is the multicellular eukaryotic cells. Right. Next, I'm coming to prokaryotic over, eukaryotic over, then finally I'm coming to archaea. In between the bacteria and fungi is called archaea. So archaea can be spherical or rod shaped or spiral, low, rectangular or irregular in and unusual flat square shaped species that also available in the salty lakes and it has been discovered from many hot springs. Some exist as a single cell, the other form filamentous or the clusters. So until the 1970s, this group of microbes was classified as bacteria. So after 1970, it classified as archaea. So many archaea have been found in the living extreme environments, for example, high pressures, salt concentrations, or high temperatures. So these type of organisms are called extremophiles. Extremophiles. So their cell wall differs in the structure from the bacteria and is thought to be more stable in the extreme conditions. Next, I'm coming to some molecular techniques which are commonly involved in the field of molecular biology. So, expression of cloning, polymerase chain reactions or PCR, gel electrophoresis, and macromolecule blotting, for example, southern blot, northern blot, western blot, and eastern blot, and DNA microarrays. Next, the allele specific oligonucleotides. The last one, antiquated technologies. Now, let us see one by one. What is the expression cloning? So, one of the basic technique of the molecular biology to study the protein function and its expression of cloning. So, in this technique, the DNA coding for a protein of interest is cloned and into the plasmid. So, in this technique, one more time I'll repeat, in this technique, the DNA coding for a protein of interest is cloned into a plasmid. So, this plasmid can be inserted into either a bacterial cell or animal cells. Next, I'm coming to in continuation of the expression of cloning. So, introducing DNA into the bacterial cells can be done by the transformation, conjugation, or by transduction methodologies. So, introducing DNA into eukaryotic cells, such as animal cells, by physical or chemical means is called transfection. So the plasmid may be integrated into the genome, resulting in a stable transfection, or may remain independent of the genome is called transfection. Next, I'm coming to, in either case, a DNA coding for a protein of interest is now inside a cell, and the protein can now be expressed. So the protein can be tested for the various enzymatic activities under a variety of situations or conditions. So the protein may be crystallized, so it is a tertiary structure, can be studied. Or in the pharmaceutical industry, the activity of new drugs against the protein can be studied. Next, PCR, polymerase chain reaction. It is extremely versatile technique for copying the DNA molecule. So, in brief, polymerase chain reaction allows a single DNA sequence to be copied. That means millions of times, or altered in predetermined ways. We can say, for example, the PCR can be used to introduce the restriction enzyme sites or to mutate. What is mutate? To change the particular basis of DNA molecule. So the latter is a method of referred to as the quick change method. So PCR has many variations like RT-PCR, that is reverse transcription for amplification of RNA, and more recently the real-time PCR, that is qPCR, allow for quantitative measurement of DNA or RNA molecules. Next, I'm coming to the fundamental technique of biotechnology is called gel electrophoresis. The fundamental most fundamental technique in the field of biotechnology. So the basic principle is that DNA or RNA and proteins can all be separated by means of an electric. So in the agarose gel electrophoresis, DNA and RNA can be separated on the basis of the size by running DNA through an agarose gel. So as usual, the proteins can be separated on the basis of the size by using an SDS phase, sodium dodecyl sulfate polyacrylamide gel electrophoresis, or on the basis of the size and the electric charge by using what is known as the gel electrophoresis. That is 2D gel electrophoresis. Next, I'm coming to the blotting technique. There are 4 different blotting techniques commonly used in the field of biotechnology. Number 1, southern blotting. So it is named after its inventor, the biologist Edward Southern. So it's the method of probing for the presence of specific DNA sequences with the DNA sample. So the DNA samples before or after the restriction enzyme digestion are separated by the gel electrophoresis and then transferred to a membrane by blotting through the capillary. So the membrane, capillary membrane, is then exposed to a labeled DNA probe that has a complement base sequence to the sequence on the DNA of the infant. So southern blotting is less commonly used in the laboratory science due to the capacity of remaining techniques such as PCR to detect the specific DNA sequence from the DNA samples. Next, I'm coming to the second blotting technique is called northern blotting. The northern blot is used to study the expression patterns of a specific type of RNA molecule. Southern blotting, DNA molecule, whereas northern blotting, RNA molecule. So the RNA molecule has the relative comparison among a set of different samples of the RNA. So in this process, the RNA is separated based on the size and it is then transferred to the membrane that is then probed with a labeled complement of a sequence of inputs. Next, the results may be visualized through a variety of ways depending on the label used. However, most result in the revelation of bands representing the sizes of the RNA detected in the sample. So this procedure is commonly used to study when and how much gene expressions is occurring by measuring how much of that RNA is present in the different samples. So it is one of the most basic tool for determining at what time and under what different conditions certain genes are expressed in the living tissues. Next, I'm coming to the Western blotting technique. In the Western blotting technique, proteins are separated by the size. In a thin gel sandwich between two glass plates. So in the technique known as SDS phase, PAGA, sodium dodecyl sulfate polyacrylamide gel electrophoresis. So the proteins in the gels are then transferred to the nitrocellulose or nylon or some other supporting membranes. So this membrane can then be probed with solutions of the antibodies. So the antibodies that specifically binds to the protein of interest. It can then be visualized by a variety of techniques, including the colored products, auto-radiography. So using the Western blotting techniques allows not only detections, but also the quantitative analysis of the product. Next one coming to Eastern blotting, the last category of the blotting technique. Eastern blotting technique is to detect the post-translational modifications of proteins. So the proteins blotted on the nitrocellulose membrane are probed for modifications using the specific substance. So further, the combinations of these techniques produce such terms as southern western blottings, that is protein and DNA hybridization. So north-western to detect the protein RNA interactions, and far-westerns protein interactions, of which are presently found in the available literature. Next, I'm coming to DNA arrays technologies or DNA arrays techniques. So a DNA array is the collection of spots attached to a solid support, such as a microscope slide, which each spot contains one or more single-stranded DNA oligonucleotide fragment. So underline single-stranded DNA oligonucleotide fragment. So each spot has a DNA fragment molecule that is complementary to a single DNA sequence that is much similar to the southern blotting technique. And then the variations of this technique allows the gene expression of an organism, particularly the stage in development to be modified. That is called expression profiling. So in this array technique, the RNA in a tissue is isolated and converted to a labeled cDNA. So this cDNA is then hybridized to the fragments on the array, and the visualization of the hybridization can be done. Since the multiple arrays can be made with exactly the same position of fragments, they are particularly useful for comparing the gene expression of two different tissues, such as healthy and cancerous tissues or healthy and diabetic tissues. So one can measure what genes are expressed and how the gene expression changes with the time or with other factors. So arrays can be made with the molecules other than DNA. For example, an antibody array can be used. Antibody array can be used to determine what proteins or what bacteria are present in the blood samples. So it's a detecting techniques for the presence of absence of bacteria in the blood sample. Next, I'm coming to the antiquated technologies. What is antiquated technologies? So in the field of molecular biology, and there is a lot of procedures and techniques continuously being developed and other technologies abandoned. Now, for example, before the advent of DNA gel electrophoresis, that is agarose gel electrophoresis or PAGE, the size of the DNA molecule was typically determined by the rate of sedimentation in sucrose gradients. Underline rate, R-A-T-E, rate sedimentation in the sucrose gradient. It's a slow and labor-intensive technique. It need much expensive instrumentation compared to sucrose gradients. The viscometry was commonly used. So aside from the historical interest, it is often worth knowing about the older technology as it is occasionally useful to solve the another new problem for which the newer technique is inappropriate. So next I'm coming to the allele specific oligonucleotides. Allele specific oligonucleotides is a technique that allows the detection of single base mutations. Underline single base mutations without the need for PCR, polymerase chain reaction, or gel electrophoresis. Very short, approximately 20 to 25 nucleotides in the length. Labeled probes are exposed to the non-fragmented target DNA. So, as usual, the hybridization occurs with a high specificity due to the short length of the probes, and even a single base change will hinder the hybridization. So, the target DNA is then washed and the labeled probes that they didn't hybridize are removed. So the target DNA is then analyzed for the presence of the probe through the radioactivity or fluorescence. So in this experiment, as in most molecular biology techniques, a control must be used to ensure the successful experimentation. So the Illumina methylation assay, IMA, Illumina methylation assay is an example of a method that takes the advantages of the technique to measure one base pair differences in the sequence. Next, I'm coming to the DNA sequencing. So there are two different methods of DNA sequencing. One is Maxwell-Gilbert method, one is called Sank's. So both methods are most commonly used in different part of the world. So here I'm giving you the main points of the DNA sequencing. DNA sequencing is the process. What process? The process of determining the sequence of nucleotides within a DNA molecule. So every organism, the DNA consists of a unique sequence of nucleotides. So determining the sequence can help the scientists compare the DNA between the which can help show how the organisms are related. So the knowledge of the sequence of a DNA sequence has many uses. First, it can be used to find the genes. The segments of DNA that code for a specific protein or a specific phenotype. If a region of DNA has been sequenced and it can be screened